Hi, my name is Kendra, and today we'll be learning about the cell cycle and all of its stages. Cells divide because what the surface area is able to supply is not enough for the demand. Big cells demand a lot, but supply is slow because surface area is small. Demand will increase as the cell grows and surface area to volume ratio decreases. So what that means is when cells keep growing, the surface area will decrease and the demand for the area is still there, so they want to keep dividing and dividing. Some essential vocabulary is the cell cycle, the big picture. The cell cycle is a series of events that cells go through as they grow and divide. Centrioles are made up of 9 triplets and microtubules. They are a part of the cytoskeleton and help support the cell division. Spindle fibers. They are elongations of the centrioles and form the mitotic spindle. Chromatids are one of the 2 identical sister parts of a duplicated chromosome. The centromere is the area where the chromatids of the chromosome are attached. Interphase, the first step in a cell cycle. This interphase is broken down into 3 steps, G1, S phase, and G2. Cell cycle, where the cell grows and performs normal activities in G1 and S phase, synthesis occurs and the DNA is replicated. In G2, the cell prepares for mitosis and organelles and molecules required for cell division are produced. This is the period between cell divisions. In interphase, the cell looks pretty normal. This is the nucleus, the nuclear envelope surrounds it, and mitosis cell division. It is broken up into 4 stages. That is prophase, metaphase, anaphase, and telophase. This process occurs when 2 identical nuclei are created before cell division. Prophase. This is the first step in mitosis. The cell will prepare for nuclear division by breaking down the nuclear envelope. The chromosomes will condense and the mitotic spindle will form. Centrioles will also move to the opposite sides of the cell. So these are the chromosomes, and the nuclear envelope is surrounding it. However, it is dissolving, so it's just breaking down. These are the centrioles. They will be at the poles of the cell throughout the whole process, and they are connected to the spindle fibers. Metaphase. Chromosomes line up in the middle, and mitotic spindle connect to the centromere. These are the chromosomes still, and the centrioles right here, and this is the mitotic spindle. They are connected to the chromosomes, and soon they will break them apart. Anaphase. Chromatids separate into individual chromosomes. Sister chromatids will move towards the poles of the cell from spindle fibers shortening. So the spindle fibers will shorten, and then they will break the chromosomes into chromatids. That is when a chromosome breaks into 2. The centrioles will still be at the poles of the cell. And then telophase. Telophase is when cells start dividing, and chromosomes will reach the end of the cell and lose their shape. Spindle fibers will start to dissolve, and the membrane begins to reform. So, when the cell starts to divide, a cleavage furrow begins to form. And the spindle fibers will continue withdrawing, as they are still connected to the centrioles. The nuclear envelope will surround the chromatin, which is the chromatids, but not chromatin. Then there is cytokinesis. This is when the cell completely divides. The cytoplasm will pinch in half, and this division of the cell will create 2 daughter cells. So it will look like interphase, but just 2. There is the nucleus, the nuclear envelope, the plasma membrane, and then 2 identical daughter cells are left. The amount of time spent in each phase will differ from each other. Interphase takes most of the time of a cell's life, more than 50%, so more than half. Then it is followed by prophase, metaphase, anaphase, and then telophase. And that is the cell cycle.